Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel, Pharma World. Today's topic is cleaning validation strategy for API manufacturing plants. Today's video focuses on strategy for optimal cleaning procedures, analytical methods, and the validation requirements. Let us move on to the equipment cleaning. There are two types of equipment cleaning in API manufacturing. They are for dedicated processes and for multi-purpose processes. In dedicated process, equipment cleaning is relatively less stringent. Section 12.70 of ICH Q7 prescribes that cleaning validation should focus on potential buildup or carryover of impurities from one batch to the other. It may not be required to validate earlier steps of the process, as there could be several further steps in which the impurities could be washed away or removed by other extractions or turning over the impurities as derivatives and removed from the process. In the dedicated process, normally there will not be any unknown new impurities. Multi-purpose processes do have different chemistries, various other potential impurities. So cleaning procedures should be critical to ensure that the contaminants are within the specified limits. Cleaning of process equipment is as important as the production process. The purpose is to check the suitability of the equipment for next stage of the process. This point is very important. Unless the cleaning validation, including the analytical method validation, the process validation is incomplete. So it is necessary to ensure cleanliness of the equipment for the next stage of the process or the next process. Purpose of cleaning validation is to establish the limits for the contaminants. Process validation is incomplete without performing the cleaning validation. Cleaning validation should follow the process validation. Clean until clean approach is not acceptable. This approach is also not practical. Regulatory agencies will not accept this approach. As a GMP also, this is not the correct approach. Achievable target should be fixed for the contaminants in the equipment cleaning operations. The equipment cleaning should meet this requirement. Achievable targets are derived based on the dosage criteria, toxicity, or general limits. Ideally, when there is a changeover from one API to the other, where there is dosage data available, these criteria should be selected. But in certain cases, the derived limits may be too high and the values look absurd. There is a capping procedure. If the derived figure is too high, it may be capped at a certain limit, like not more than 2000 ppm. When there is a change from an intermediate to another intermediate, toxicity data may be considered. In such cases, there will not be any dosage criteria. The general limits may be set between 5 ppm and 100 ppm depending upon the nature of the product. 10 ppm in API manufacturing is frequent. The limits should be calculated based on scientific rationale. Brief information on calculation of macro values is provided in the last slide. Let us see the aspects of cleaning now. Maximum allowable carryover, that is MACO, should be calculated for each piece of equipment. As the first step, this should be done. Most stringent, that is the worst case limits, should be assigned for release of the equipment for manufacturing of the next product. Worst case means the lowest macro value among all the shared processes. If the lowest is selected, it will be suitable for manufacturing of any product in that shared list. Acceptance criteria for cleaning of equipment include no visible residue, swab limit, and rinse limit. When one's MACO is done, the acceptance criteria should include these items. Good lighting is necessary to check the no visible residue in the equipment. Lighting plays a more important role while using glass-lined equipment in the process. The glass lining is generally dark in color. Stainless steel equipment are shiny and easily checked. Swab technique is more suitable for sampling of hard to reach locations of the equipment. Samples include deposits in remote corners which cannot be checked visually. Also, by this technique, residues that are dried out or are insoluble can be sampled by physical removal. Rinse sampling is indirect method. This gives information about overall equipment cleanliness. Total carryover is sum of contamination from each shared equipment. This is important point to note. The overall set limit should be not more than the sum of residues from all shared equipment. Let us see the strategy for dedicated equipment. Batch to batch cleaning is by visual inspection, no visible contamination. This is only physical removal or de-dusting of equipment free from previous batch. As explained above, it is necessary to have proper lighting to check the physical cleanliness of the equipment. Scheduled cleaning depends on the trends of potential buildup of impurities. Scheduled cleaning means carrying out detailed cleaning with a definite frequency. This is generally based on the number of batches per campaign. The focus should be on the increase of contaminants beyond limits. In certain cases, time limits like once in a month or so can be set for detailed cleaning. Detailed cleaning should contain the details of detergents used for removing contaminants, removal of detergent after cleaning, disassembling and reassembling of parts should also be part of the cleaning procedure. The alert limits for cleaning schedule must be derived based on the results of statistical evaluation of the available data. Six Sigma approach may be adopted for alert limits. Plus or minus 3 sigma should be for final product specifications, whereas tighter 2 sigma may be set for alert limits. So the cleaning schedules may be revised if there is any trend beyond 2 sigma values for the contaminants. Cleaning schedules may be relaxed if there is a positive data indicating full control on impurities within a very narrow and lower range. If the data is very narrow and there are no signs of going out of trend, the cleaning schedules may be relaxed. This saves on resources also. On the contrary, the cleaning schedules may be tightened if the data is not favorable. In such conditions, it may also be necessary to investigate revising the cleaning methods to be within the OOT set limits. Let us see the strategy for multi-purpose equipment. A detailed risk assessment report must be made for cleaning of equipment used for multi-purpose. Useful tools like FMEA, FMECA, HACCP should be used to get a comprehensive risk assessment report. Grouping of equipment depending upon commonality in cleaning elements is a better and optimal approach. Common cleaning elements include the use of common detergents, common procedure, solubility, etc. Identifying the worst case among each group is the next step for an optimal approach. Among all methods, identifying the worst case is much more economical. Good logic and scientific approach with a cost benefit angle would lead to an optimal strategy. There is no hard and fast rule for these. Good scientific and logical investigations into the risk is necessary. All minor and major points should be tabulated and evaluate the potential risk for each point. The focus point for the risk assessments are provided in the next slide. The risk assessment should include the information from the following: toxicological or pharmacological activity of the previous product, maximum daily dosage of following product, microbiological growth, batch size of the following product, solubility, experience difficult to remove previous product. All these aspects should be addressed one by one. Let us see a typical product changeover scenarios. The risk assessment should recommend 3 levels of cleaning. They are: level 2, carryover of the previous product is critical. Cleaning should be done to meet the stringent carryover limits are met. Level 2 is critical. There is a potential for impurities beyond limits if the cleaning procedure is not comprehensive and validated. Generally, cleaning validation is carried out with 3 consecutive cycles. Level 1 is carryover of the previous product is less critical. Cleaning should be done to meet a less stringent carryover limits as required for level 2. This is also critical to the extent of achieving less stringent limits. Level 0 is gross cleaning if the previous product is not critical. Physical cleanliness is adequate in this
level of cleaning. The cleaning procedure levels are as follows. Let us consider process A. Let us consider that there are three stages in process A. They are A minus 3, A minus 2 and A minus 1 before becoming crude API. Cleaning of equipment from earlier stages of A minus 3 to next stages of A minus 2 and or A minus 1 level 0 is adequate. So cleaning of equipment from earlier stage of A minus 3 to next stage of A minus 2 or A minus 1 may adopt level 0. There are some more processing steps to remove the impurities, hence level 0 may be justified here. Cleaning from stage A-1 to crude API or pure API, level 0 or level 1 may be suitable depending upon the criticality. As the process is approaching crude API stage from penultimate stage of A-1, where the API molecule is formed, chances of contamination from the previous stage should be controlled. So, depending upon the criticality of the process, generated impurities and the capability to remove those contaminants, it may be necessary to have level 0 or level 1. But when the changeover is from A-3 or A-2 stage to crude or pure API stage, it is necessary to have level 1 or level 2 cleaning. In this case, the changeover is from earlier stages of the process to the crude API stage. This is critical. There is a potential for remnants of contaminants or impurities into crude API. In such cases, this is the strategy. Same strategy may be adopted for any other process. So, any process, this is the same strategy. Change or cleaning from one earlier stage of one process A to another earlier stage of another process B, depending upon the criticality, either level 0 or level 1 may be adopted. This strategy is selected because the processes are two different processes, A and B. The chemistry and impurities may be different. So, it is necessary to evaluate through a risk assessment on the levels of cleaning. Changeover cleaning from one intermediate stage of one process A to crude API or pure API stage of another process B, it is recommended to go for cleaning level 2. This is a critical situation. There are two processes involved and the changeover to an earlier stage of one process to crude API stage of another process. All physical operations of changeover cleaning from one process A to another process B should always be cleaning level 2. This is an obvious choice. Physical operations include drying, milling, sifting, blending, etc. This is the final product for marketing. There are no further processing steps to remove contaminants. Any traces of contamination would end up in the final product rejection. Let us look into validations. For level 2 cleaning, both analytical method and cleaning methods should be validated. This step is ultimate for cleaning validations. All steps of cleaning should be validated before the equipment is released for manufacture of any product. The swab and rinse limits should be met. The validation project should also include the detailed training requirements. The analytical methods should be validated. Limit of detection and limit of quantitation values must be established carefully as it is necessary to determine ppm levels of impurities. UPLC methods may be developed for evaluation of cleaning samples. UPLC is much faster than HPLC. Alternatively, UV methods may also be developed as standby provision, but it is necessary to establish equivalency to each other technique. For level 1 cleaning, lower acceptance criteria may be set as there are more steps that could remove the contaminants. Same strategy for this too. The only difference could be relaxed specification limits. Based on the risk assessment report within the same synthetic process or in different synthetic process, validation should be executed. Detailed risk assessment will help in this. Risk assessment may include information from various aspects. Aspects as detailed in the earlier slide should be considered for risk assessment. Let us see the approach. To achieve optimal or minimal validation requirements, bracketing and worst case rating should be done. If there are 5 equipment trains shared for 10 processes, it may be necessary to have 50 validations to accommodate all the processes, but it is not practical to do that way. By bracketing procedure, the substances could be grouped. Bracketing is achieved by grouping on common factors among the processes. In each group, worst case should be selected. So again, in each group, the worst case should be identified. Validation should be done for the worst case situation. However, the selection of worst case should be based on scientific rationale. So if the worst case is validated, it is considered that the system will work for all other situations. Worst case may be established depending upon hard to clean, solubility, toxicity, and therapeutic dosage. This is how the worst case is selected. Details of maximum allowable carryover macro calculations for determining the amount of residue were discussed in earlier video program titled Cleaning Validation dated September 2021. You may please refer to this video for more details on macro calculations and different approaches for macro calculations. Analytical method should be validated as per USP chapter 1225. There are several validation parameters including LOD and LOQ in USP chapter 1225. I hope that this brief information on various strategies for cleaning validations is useful as a startup. There is a useful guide titled Guidance on Aspects of Cleaning Validation in Active Pharmaceutical Ingredients Plants dated December 2000 and updated guide dated February 2021. Thanks for watching. For more videos, please do subscribe, like and share. Thank you.